Hi everyone. My name is João, as it was said. Actually, it's quite nice to be here. When I first started studying bioinformatics, or when I started doing bioinformatics, because I'm a biologist, just to keep the trend of the first day, actually, my supervisor told me, well, do you know programming? And I said, yeah, I know some Pascal. And he said, no, just Google Python and Google biology and see what you get. And Biopython showed up. So this was in 2007. I was kind of a lurker for a very long time, probably annoying Peter with some nasty questions about, yeah, bioinformatics that I didn't know anything about. And over the years, I became a bit more involved. And yeah, so this year I got the honor of coming here and presenting the updates that of what has been happening in the community. So I don't have Twitter. You cannot abuse me on Twitter. You can abuse me orally, like you just ask questions if you want. So that's kind of how it goes. I'll just introduce a bit what the Biopython project is all about in the end, because I don't know if most of you actually know it. I hope you do. So if you go to the webpage, you actually see that it's one of the BioStar projects under the Open Bioinformatics Foundation umbrella. And basically, it's a set of tools for biological computation written in Python. So it kind of take it from the name, right? If you keep on reading the description, it also says it is a distributed collaborative project. And this is actually quite interesting because, so it was only now, the day before yesterday, that I actually put faces to all the names that I've been seeing for the last 7 years. So it was quite interesting. So if you look at if you go on GitHub where we have all the code, you see that the development started in late 99. And we just actually passed the milestone, I would say, of 10,000 commits on GitHub. I don't know if this was celebrated with beers one of these days, but it should. And these have been made by more than 91 contributors. So it's quite a diverse, actually, set of contributors, of people contributing code to the project. And if you just see who are the 10 most, well, first of all, photogenic people, and also the ones that contribute the most, this is the set of people that you come up with. And the interesting part is, we've been an active participant in the Google Summer of Code programs and for the last 6 years. And it's nice to see that we actually have a good retention rate. So of all these top 10 people, actually you see that half of them actually are previous Google Summer of Code students. And I was also one of them, but unfortunately I'm ranking number 26. So I don't know what's going on there. And so somebody was saying something about career progression in open source. So one of the first students, so Eric Talovich, who gave this talk a couple of years ago, he started as a student in 2009. He became a mentor to me in 2010. He's been a mentor also for all these years, and now he's the administrator also from, I think, the OBF part for the Google Summer of Code. So if there's a career progression in open source, this is one path you can actually follow. So now for the update, actually, what has been happening in the code. So there was one release since last POSC. Doesn't mean that not a lot of it was done. Doesn't mean that there was not a lot of stuff done, actually. It was kind of the opposite. So these are all the modules that were kind of updated in between. And I also am going to just plug in a bit the changes that are planned for the next release, which is due quite soon, actually. So starting with the BioKeg and biographics. So KEG is the Kyoto Encyclopedia of Genes and Genomes. Basically, it stores a lot of pathway information and a lot of metabolite information. And what has been done was actually, it was added support for their RESTful API. So now it's possible you can just make a call to their API from the library, and you can use it to retrieve any information on the database, on the information, on the contents. And this was also coupled with the graphics module of Biopython, which allows you to actually draw stuff using Matplotlib or ReportLab. And so it was also added support for these pathways. So they have this markup language, and then you can download it as well, and then it can be used to actually draw fancy things like this. So this is just one metabolic map that you can just download from the webpage with a quite simple piece of code now. And also you can include it as well in a Python notebook. So for education, this is also quite an interesting approach. These were contributions of again, like very different people that have been contributing to the code. And if you go to this webpage, you see not only the code and some examples, but you also see a full IPython notebook telling you the several possibilities that you have of interacting with the API. So moving a bit on, so there's always this need to have updated data in the package. So there was just NCBI released another genetic code table. So we added that, and this was also updated, like in all the genetic code tables and the translation functionalities. So although I'm a structural biologist, so sequences are just, the first thing I look at, just a string of letters, then I'm actually curious about how they actually play in space. But so one of the core, I would say, parts of Biopython is the sequence parsing modules and the alignment parsing module. So these one actually, they were one of the focus of all these updates in this last version. So there were some additions to actually support file names and index files in a relative way, and also an extension to one of the format parsers that allows you to expose actually the raw data from the file, and then this, if you actually know what you're doing, then you have much more powerful way of accessing the raw information. And also, so last year, one of the Google Summer of Code students we had was actually involved in updating the sequence module to actually make it the indexing and the parsing lazy, so that you can actually access like very large genome files and you don't have to load them all in memory. And this is, of course, much better than just having to put the whole thing in memory. And I think one important thing is that it's fully compatible with the existing parsers already, so there is no breaking in compatibility, which is kind of a trend that we also see in Biopython. There was also, so I think 3 years ago or 2 years ago, so Bo started also as a summer of code student, and he was involved in creating from scratch, I think, the Search.io module. So it's a unified interface for sequence search, so it's for BLAST, for Hammer, for all these kind of softwares. And this was also updated now during the last year. And again, it got the same update as the sequence.io regarding the file names, and it also, there's an improved retrieval of the hits, so you can actually access the hits in your search results based on alternative IDs, not only the primary ID of the hit. And during the Code Fest, so 2 days ago, there was actually several upcoming fixes and new parsers. He was working actually quite heavily on this all the time. So it's actually quite interesting to see what's coming out. And also quite interesting to see also the first Code Fest, so that's also cool. So an extensive actually rewrite, and this was actually, I think, quite good, was the melting temperature calculation, so for sequences, both DNA and RNA. So somebody actually took time to actually rewrite the whole thing. So first of all, they fixed some errors that were there for RNA temperature calculations, the melting temperature calculations, and they added 2 other methods for this. So this is much more generic now, the way it is. And driving home for the last bit, so the PDB module, so PDB stands for Protein Data Bank, but it also stands for the format that the Protein Data Bank used to actually have their data on. So the structures that you see from proteins, from molecules are stored, or were stored in this PDB format. And this is actually now being deprecated, or is going to soon be deprecated in favor of this MMCIF format for reasons that are coffee breakable. So what happened here, so this is actually not in this release, but it's going to be included in the next one. So there was a different way of calculating similarities between structures. So when you want to compare structures, you have to kind of superimpose them on each other. So there are 2 normal ways of doing this. So there was one, which was singular vector decomposition, and somebody added basically a quaternion-based approach, which has advantages over the previous one. Somebody also extended the DSSP. So DSSP is the Dictionary of Secondary Structure in Proteins, something like this. So it's a program that's been cited like 15,000 times since the 80s or 70s. And it's used basically to extract secondary structure information and hydrogen bond information from the structure. So this was also included. And during the Code Fest, I was also working a bit on improving our MMCIF parser. So just a little tweak made it 10 times faster. So instead of taking 200 seconds, now it takes 20. So that's a very good thing. Still needs to be, tested thoroughly though. And we also corrected a few long-standing issues together with some new collaborators that we managed to actually draw in. And finally, something I found actually quite funny when I was looking at latest commits. So not all commits are about having amazing functionality. So some commits are just about thanking themselves. So this was a fun one that we found. So I think Carlos, he went basically over the entire code base, and he was just pepating the entire code base. So he's currently like the 11th top contributor on GitHub just because of this, which is good because he was cleaning the whole thing. So beyond the code, there are 2 things that I would like to just plug in. So the first one is, and I'm sorry to break the Docker chain here again and bring it up. So Tiago has actually been working on implementing some Docker containers that include Biopython, and they can be used if you just want to deploy it quite quickly, or also for education purposes. So he also has containers that contain the IPython thing in there. And you might ask like, what's the problem with all the dependencies we have? Well, most of them actually come probably from the structural module that have all this code written in Fortran and in C from the 70s and 80s, and these things are, from experience, a pain to install. So this was actually quite nice to have around. So it does include all the dependencies because some of them are not freely available. So that's the bad thing. And lastly, so Tiago was supposed to go to SciPy to give kind of the same presentation as I'm giving, but he got denied the visa because it is in the US. But meanwhile, he got some happy news. So he published a book on using Python for bioinformatics. So this not only covers Biopython, but it covers many other things. Covers NumPy, SciPy, optimization methods. So using Cython and Numba, for example, if you really want to extract all performance you want from your code. And the good thing is all the example codes that are in the book are available online on GitHub. And he will, at least he committed that he's going to maintain them and correct them as things actually show up. And the book is also available if you want to get it. So just to save time for the next person, I'm done. And I'd like to acknowledge, so first of all, the people that allowed me to be here and to be part of these projects, all the contributors that actually made the project what it is, and of course all of you for putting up with me for roughly 10 minutes. So thank you.